Hi all, welcome to Physio Exam Buddy. In today's video, we will discuss the role of kidneys in the regulation of the plasma pH. It's an important concept in the acid-base physiology and a frequently asked exam topic. Let's understand the key mechanisms clearly and make this topic easy to revise for your exam. Kidneys, they regulate the H plus ion concentration in the extracellular fluid mainly by three mechanisms. The first one is by the secretion of H plus, which is also known as acidification of urine. And the second is by reabsorption of the bicarbonate, and third by production of new HCO3 or bicarbonate. Out of this, the first two steps, that is both the secretion of H plus as well as the reabsorption of HCO3 minus, takes together. So it can be explained in the answer also together. First, we will see the secretion of H plus as well as reabsorption of HCO3 minus. Both the reabsorption of HCO3 minus and the excretion of H plus, they are accomplished through the H plus secretion by the tubules. And this H plus secretion occurs in all parts of the tubule except the thin ascending limb as well as the descending limb of loop of Henle. The secretion of H plus as well as reabsorption of the bicarbonate. Out of this, the 80% occurs in the PCT, 10% in the thick ascending limb of loop of Henle, and the remainder in the distal tubules as well as in the collecting ducts. First of all, we will see the secretion of H plus. The mechanisms involved mainly include a secondary active transport, mainly in the proximal tubules, thick ascending limb of loop of Henle, as well as in the early distal tubule. Whereas in the late distal tubule, mainly in the intercalated cells as well as in the collecting tubules, the secretion of H plus occurs by means of a primary active transport. First of all, we will see the secretion of the H plus along with the reabsorption of the bicarbonate occurring in the proximal tubules. And this is the diagram you have to draw while explaining this in the answer. It is usually our carbon dioxide. It will diffuse into the tubular cells, or it can also be formed by the metabolism inside the tubular epithelial cells. So inside the cells, the carbon dioxide combines with water in the presence of the enzyme carbonic anhydrase to form H2CO3. This H2CO3 is a highly unstable compound, so soon it will dissociate into H plus as well as HCO3 minus. And this H plus, it will be secreted into the tubular lumen through our apical membrane by a counter transport mechanism, which is an example for secondary active transport, that is sodium hydrogen counter transport. And this sodium, which moves into the cell down a concentration gradient, which is established by our sodium potassium ATPase, which is present in this basolateral membrane. And the hydrolysis of this ATPase, it will provide the energy for this H plus secretion. Along with this, the HCO3 minus, which is formed from the carbonic acid, it will move downhill across the basolateral membrane into this renal interstitial fluid as well as into the peritubular capillary blood. And each time an H plus is formed in the tubular epithelial cells, along with that, a bicarbonate is also formed and released back into the blood. Now we will see what happens to the secretion in the distal tubules as well as in the collecting ducts. Here the mechanism is mainly by the primary active transport with the help of an H plus ATPase as well as an H plus K plus ATPase, both of which are located in the apical membrane of the tubule, which is facing the tubular lumen. And the energy required for this is also derived from the breakdown of the ATP into ADP. In the distal tubules as well as in the collecting ducts also, the dissolved carbon dioxide combines with the water to form H2CO3, which soon dissociate into bicarbonate as well as the H plus. And this H plus will be secreted into the tubule either via H plus ATPase pump or via the H plus K plus ATPase pump, both located in the apical membrane of the this tubular epithelial cells. And here the main difference is that here the H plus will move across the luminal membrane by means of an active H plus pump instead of the counter transport. That is the only difference between the proximal tubule as well as the distal tubule in the secretion of the H plus. And the pH of the glomerular filtrate is usually 7.4, whereas the pH of urine is equal to 6, and the minimum pH possible is the 4.5. And the last mechanism by which the kidneys regulate the acid-base balance is by the production of the new HCO3. That is, when an H plus is secreted in excess of the HCO3, only a small part of the excess H plus can be excreted in the ionic form, that is H plus form in the urine, because the minimal urine pH is only about 4.5. And whenever there occurs an excretion of large amount of H plus, this excess amount of H plus will be excreted by combining this H plus with certain buffers. And the most important buffers involved in the excretion of H plus whenever the body excretes large amount of H plus is by phosphate buffer as well as the ammonia buffer. And other weaker buffer systems can also be involved in the production of bicarbonate like urate as well as citrate. So in this video, we will be seeing only the main two buffer systems, that is the phosphate buffer system as well as the ammonia buffer system. So along with the phosphate buffer system, you have to draw this diagram. And the main phosphate buffers include HPO4 as well as the H2PO4. So once all the bicarbonate is reabsorbed, the excess H plus which is generated in the body will combine first with this HPO4 and as well as with the other tubular buffers. And when this H plus combines with the sodium salt of this HPO4, it will form H2PO4, and it is usually excreted as sodium H2PO4, carrying with it the excess H plus ions. And whenever an H plus is secreted into the tubular lumen combines with a buffer other than HCO3, the net effect is an addition of a new HCO3 to the blood. And the next buffer system which helps in the excretion of the excess H plus is the ammonium buffer system, which mainly include ammonia NH3 and the ammonium ion, that is NH4 plus. This ammonium ion, it is mainly synthesized from the glutamine, and the glutamine is delivered to the kidneys and it is transported into the epithelial cells of the proximal tubules, thick ascending limb of loop of Henle as well as the distal tubules. Now we will see what happens in the proximal tubules for the excretion of the H plus because of this ammonium buffer system. So we have seen that this ammonium ion, it is formed from the glutamine. So in the proximal tubular cells, each glutamine is metabolized. It will form two NH4 plus ions as well as two HCO3 minus. And this NH4 plus, it is secreted into the tubular lumen by a sodium counter transport. And this HCO3, it will be transported across the basolateral membrane along with the reabsorbed sodium taken up by the peritubular capillaries. So for each molecule of the glutamine, two ammonium ions will be secreted into the urine for the excretion of the H plus. Along with that, two HCO3 minus is also reabsorbed into the blood, which will constitute the new HCO3, or this is the new generated HCO3. Now we will see what happens in the collecting tubules. Here, the H plus is secreted into the lumen. It will combine with the NH3, that is the ammonia, to form NH4 plus, and it is excreted. And this collecting duct, one of the peculiarity of this collecting duct membrane is that it is permeable to this NH3 but less permeable to this NH4 plus. So what happens to the NH4 plus is that it will get trapped in the tubular lumen and is eliminated in the urine. So for each NH4 plus is excreted, a new HCO3 is also generated and it is added to the blood. So that is about the renal regulation of the plasma pH, and along with that you have to write the applied physiology, and the applied physiology here is the renal tubular acidosis. It is a condition in which the renal acidification is impaired, and there are two types of renal tubular acidosis. First one is proximal renal tubular acidosis and the second one is distal renal tubular acidosis. In the proximal renal tubular acidosis, the defect is in the proximal tubule H plus secretion. So there will be decrease in the reabsorption of the filtered bicarbonate also, and the decreased plasma bicarbonate will cause metabolic acidosis. And the main causes for this proximal renal tubular acidosis are Fanconi syndrome as well as administration of carbonic anhydrase inhibitors. Next is the distal renal tubular acidosis. Here the defect occurs in the distal tubule H plus secretion, and it is due to the defect in intercalated cells. So that's about the renal regulation of the plasma pH. Thank you.